Hi, everyone. Arsalan Arif here with Endpoints News, and thanks for joining us today for Applying Machine Learning to Increase Clinical Trial Efficiency, a Regulatory Journey. We're sponsored by Unlearn AI, and I'm excited to moderate today's discussion with Charles Fisher, the founder and CEO of Unlearn AI. Unlearn is the only company creating twin RCTs, which combine AI, digital twins, and novel statistical methods to enable smaller, more efficient clinical trials. Unlearn's technology has been published in conference abstracts in scientific journals, including Scientific Reports Nature and the International Journal of Biostatistics. Unlearn is partnering with the world's leading pharma companies, including Merck KGaA, Darmstadt, Germany, and continues to have discussions with regulators. The European Medicines Agency published a draft qualification opinion for PROCOVA, a patent-pending method developed by Unlearn for leveraging historical data and machine learning to reduce sample sizes or increase power in phase 2 or 3 clinical trials. Now, if you have any questions during today's webinar, our panelist has reserved some time at the end, so just hit the Q&A button for Charles at the end as soon as you think of the question. This webinar will be available on demand tomorrow to rewatch or to share it with your colleagues. And now, Charles.
Thanks, Arslan. Glad to be back here with Endpoints, doing another webinar. And we're going to try to make this as entertaining of a presentation as we can, talking about our TwinRCT solution. Again, the title here is that we're applying machine learning to increase clinical trial efficiency. And we're going to talk about our regulatory journey, the various things we've learned along the way about how we can do that in a way that regulators will accept. Basic outline for today. I'm going to start off at the very beginning by giving everyone the take-home message. I know when I listen to presentations, I have a short attention span, so deliver the take-home message right away. Then I'm going to do just some definition about technology we call a prognostic digital twin. And then finally, I'm going to walk through the story of our journey, creating the solution, and then applying it in a way that the regulators will accept. So let's start with our take-home message. So what exactly is a TwinRCT? It is the convergence of 3 different technologies or resources. The first is historical data from the control groups of previously run trials or observational studies. So we can aggregate data in electronic format from all of these different previously completed studies in order to build large data sets of individual level longitudinal patient data. Then the second is deep learning, new machine learning technologies based on artificial neural networks that allow us to build sophisticated models from these large compendia of data. And the third are biostatistics for RCT design, figuring out how we can develop randomized controlled trials that use predictions from these deep learning models to improve efficiency and power. And so the convergence of these 3 different technologies is what we call a TwinRCT. So TwinRCTs allow us to provide clinical trials with a few different properties. First is that we are able to run these clinical trials with smaller control groups. That makes them, well, smaller overall, so they enroll faster, but also more attractive to potential participants who have a higher chance of receiving the experimental treatment. They maintain high statistical power, just like a larger RCT. They control bias, just like a larger RCT. And they produce regulatory suitable evidence for both phase 2 and 3 clinical trials. And the EMA recently published a draft qualification opinion on the statistical method that underlies TwinRCTs, in the specific case of continuous outcomes. So over the course of this presentation, I'm going to talk about how we got here and the various lessons that we learned along the way, developing the solution and then bringing it forward to regulatory review. But before I do that, I want to just define a few terms. In particular, I want to talk about this technology that I'm going to call a prognostic digital twin. So a couple of weeks ago, I was on a panel with a number of different CEOs working on technologies for clinical trials. And the moderator asked me, what does Unlearn do? And I said, well, Unlearn creates digital twins of patients in clinical trials. And by digital twin, I mean a computer model that allows us to predict the future state of health of that patient under some condition. And then the other panelists went around and they said what it is their companies do. And another CEO said, well, my company also creates digital twins. And I thought, well, that's interesting. It's interesting to be on a panel with another company that sounds like they could be competitive. But then he went on to say that what his company does is that they provide wearables or other digital biomarkers to patients that allow them to create digital twins, which he described as a detailed picture of a patient's present health. So what exactly is a digital twin? Those are completely different things. At Unlearn, we're using digital twins to think about future health, but other people are using digital twins to mean current health. So if you look at the market, people are using the same term for two completely different things. So just to make sure that it's clear, I'm going to use the term prognostic digital twin. And a prognostic digital twin, again, is going to be a prediction about the future state of health of some patient. And that's also important because it is a prediction about some individual person. You can't say just like, I want 100 digital twins. 100 digital twins of who? It really matters that the goal ultimately is to have an actual person and observe their outcome. And you're going to have their digital twin with a predicted outcome. You're going to compare those things. So we need to think about a digital twin, a prognostic digital twin of some individual person. So we can have a prognostic digital twin of Alice or a prognostic digital twin of Bob. In general, it's a prognostic digital twin of some particular person, some individual. And what we mean by that is a computer model that predicts the potential health of that person in the future under some conditions. And I know it may seem like I'm saying that sentence many, many times, but honestly, if there's one thing to remember from this presentation, it's probably this slide. A prognostic digital twin of a person is a computer model that predicts the potential health of that person in the future under some conditions. Okay, so then what is a TwinRCT? A TwinRCT is a randomized controlled trial that uses these prognostic digital twins of all of the study participants to enable clinical trial designs that have smaller control groups while still providing high power. So we want to be able to make a trial that is smaller, enrolls faster, is more attractive to potential participants, but provides all of the statistical properties that we would expect from a larger randomized controlled trial. Okay. So now I'm going to walk you through this journey of how we developed the solution and some of the lessons that we learned along the way. And to do that, I'm going to use this framework that I call the cloud. Now, I say I call it the cloud. I don't exactly call it the cloud. I learned this from a professor of mine in grad school. He actually has a TED Talk about this idea. And he's thinking about this in terms of scientific progress. So often when we talk about running experiments, we say that we're going to go from A to B. We have a hypothesis, and we're going to run our experiment. We're going to start at A, we're going to end up at B. But in many cases, that's not really how it works. In real life, we start out at A, and we start our experiments thinking that we're going to end up at B, but we instead wander around through this fog he calls the cloud. And here is where you're trying different experiments, you're getting unexpected results. In many cases, this is the deep toil of doing a PhD. And you end up sort of leaving the cloud at a different place than you predicted. So instead of going from A to B, you actually go from A to C through this long, twisted journey. And then you write your scientific paper and you say you went from A to C. You don't talk about all of these things that happen in the cloud. And that's really unfortunate, because that's the area where you learn all of the important lessons. So the cloud is where we learn all of our most valuable lessons. And I want to talk about our journey at Unlearn through this cloud and what we learned along the way. And if we think about just a high level picture of the cloud, and I'm going to go through these different pieces in detail. We started off with this idea that we could take historical control data and we could use deep learning to develop simulated control groups for clinical trials. So we could just simulate the control group. But as we wandered around through different experiments, working with biopharma partners, talking to the regulators, we learned a lot along the way and ended up at a different solution, which is uses of digital twins within randomized controlled trials. And that's the journey I want to talk about. So we're going to compare two different use cases. The first one is one in which we will create digital twins of all participants in a randomized trial. So that means we're going to have an RCT. Some patients are going to receive the experimental treatment. Other patients will receive a control. Those are randomly assigned. And every single one of those patients, both the patients in the treatment arm and the control arm, are going to create prognostic digital twins of and use them in the analysis of the trial. And that's going to enable us to design that trial to have a smaller control group. And we're going to compare that to a different concept, which is where we create digital twins of only patients who are receiving the experimental treatment. And we're going to use their digital twins, as a simulated control group in a trial that does not have a concurrent control arm, so a single arm trial. These two different types of designs are completely different. They have completely different statistical properties, and that's what we want to talk about today. So let's talk about Act 1. This is the beginning of Unlearn, 2017, 2018. And we were, in fact, interested in how we could potentially apply these new machine learning technologies to create simulated control arms. Now, at the time, there was a lot of interest in how you could use external data sets, whether it be historical control data sets or real world data sets or whatever, to reduce the size of the control arms in clinical trials. And in particular, rather than running randomized controlled trials in which half the patients receive your active treatment and half the patients receive your control, there was a lot of interest in using external control arms. So now you're going to run a clinical trial where all of the patients who enroll in your trial receive your active experimental treatment. And you're just going to compare that to data from someplace else. And this is, of course, this is done in certain indications, rare indications. It's done in phase 2 trials where you might use a placebo group from a previously run trial. Or potentially you're going to use some statistical methods called propensity score matching to pull in data from real world sources. And then people call this a synthetic control arm. The umbrella term, though, is external control arm. And so we took a very different approach to this problem. Rather than trying to take real patient data and match it into the trial in some way. So actually taking real patients from a different source, saying we'll use this group of them as a control. We thought about actually creating computer simulations of control groups. So the whole idea here is that we can take large amounts of data from historical control arms, not of one study, but of many studies. So we might aggregate data from dozens of previously completed clinical trials. We might add in observational studies. We might even add in some real world data. And then we can develop these deep learning models that people call generative models that would allow us to create simulated control groups for that trial. And in particular, it's not just like you could simulate a group of patients, which you can, but we can actually simulate this counterfactual question for individual patients. So we will use these deep learning models trained on historical data to create these prognostic digital twins of individual patients. So here's an example. We have a patient, she enrolls in the clinical trial, and at her first visit, collect all of this data at baseline. And those data are then input into a pre-trained and pre-specified AI model that creates her prognostic digital twin. And that digital twin is a prediction of how all of these different measurements might evolve over the course of the trial if she were to receive the control. And then you have the real patient, and you give them the treatment and follow her for all of the different follow-up visits and end up with an observed outcome, an observed evolution of all of these measurements over the trial. So we have an actual outcome and we have a predicted outcome for each individual patient. So here is an example of a prognostic digital twin for a patient with Alzheimer's disease that we have created. You have a lot of detailed information about that patient collected at the beginning of the study. So there are all of these different measurements at the top, like age and sex, some genetic information. Those are all sort of background measurements. Then you have all of these different measurements about that patient's cognitive states and blood tests, other things. And all of those things are measured at baseline at the patient's first visit. They are input into this pre-trained computer model, and we predict all of these other things. So we are creating a comprehensive prediction of every outcome for this patient over the course of the clinical trial. And we published some descriptions of our machine learning methods that do this. We also have a number of patent applications around different approaches for this. But the generic idea is to create these comprehensive predictions of control outcomes for individual patients. And in principle, if you could do that very effectively, then you could use it to estimate treatment effects from trials without control groups. So at the end of the study, we have the actual outcome that was observed from the real patient who received the experimental treatment. And then we have the predicted control outcome for that patient that we get from their prognostic digital twin. And if we simply subtract that predicted control outcome from the observed treatment outcome, then that should be an estimate for how effective that treatment was. And you can aggregate that information over the population to get an estimate for the average treatment effect, which is usually what we're after in a clinical trial. But there's kind of an immediate question that pops up to most people who are familiar, not just with machine learning, but statistical modeling in general. What happens if the predictions from our model are biased? So suppose that we predict that patients' outcomes on the control are worse than they really would be. Well, then when we actually take this difference between the observed and predicted outcomes, we would be overestimating the effect of the treatment. And that could lead us to potentially conclude that treatments that really don't work very well, that they're better than they are. So if our predictions are biased, then how could we trust the estimate for the treatment effect that we get out of this approach with a simulated control arm? So that leads us to Act 2, which I'm going to call confronting the regulations. So external control groups, for a variety of reasons, are not typically appropriate for most phase 3 clinical trials. They can be used in some special situations with maybe ultra rare diseases or particular indications where there's some ethical consideration around randomization, but most of the time, they're not appropriate for these phase 3 pivotal clinical trials. And you don't need to take my word for that. This comes directly out of FDA guidance E10. So the FDA says that the inability to control bias is the major and well-recognized limitation of externally controlled trials and is sufficient in many cases to make the design unsuitable. And it turns out that this statement applies to all of the methods that people have for true external controls of clinical trials. Historical control could be biased. A synthetic control arm created by propensity score matching could be biased. A simulated control arm where you simulate control outcomes with digital twins, that could be biased. So all of these things, they all have this problem of potential bias if you eliminate the control group and run a single arm trial. So we wanted to get a better idea of how FDA was thinking about clinical trial designs. What are the characteristics that they really want to know about? So we had a meeting, a critical path innovation meeting with FDA in March of 2020 to discuss their perspectives on simulated controls and novel clinical trial designs. And they said something very clear. Their question is, how often will the use of a novel clinical trial design result in a different outcome due to unknown unknowns that you cannot account for in the model? This, again, this applies to what we were doing with simulated controls, applies to synthetic controls as well. When you're using modeling, you can account for the factors that you know about, but you can't account for all of the stuff that you don't know about, can't measure, or just didn't measure. All of those potential things could lead you to have an incorrect conclusion in your clinical trial. But this also tells us the problem that we need to solve. We need to think about, we need to come up with a way that allows us to use these prognostic digital twins of participants in trials to design more efficient trials, where we can demonstrate that you don't result on average in different outcomes due to unknown unknowns, that you have the same robustness to potential confounders that you would have in a regular clinical trial. And to mitigate these potential confounders from, again, from the things that we don't know about, that we haven't measured or can't measure, there's really only one tool that we have, and that tool is randomization. So rather than thinking about just eliminating the control group of a trial and running a single arm trial, we need to find a way to put randomization back into the study. So now we want to say, okay, we're going to be combining the same historical control data, these same deep learning based methods for predicting prognosis, but we're going to include that information into novel randomized clinical trial designs. And that leads us to this last thing, the TwinRCT revolution. Thinking about that problem, how do we incorporate these predictions of control outcomes that we get from prognostic digital twins into the design and analysis of randomized controlled trials? We developed a statistical method, or it's really a family of statistical methods called PROCOVA. And these methods allow us to leverage that information within randomized clinical trials to make them more efficient, to reduce the number of patients that you need to enroll while maintaining the same power. And they do it in a way that is robust even to unknown unknowns. And again, we have published some of the theorems that describe the statistical properties of this approach in the International Journal of Biostatistics, and also have a number, actually a large number of patent applications describing various variations on this theme. And in general, PROCOVA looks like a 3-step process. In the first step, we are going to train and evaluate a prognostic model to predict outcomes on the control. And that means we're going to create a model that can create prognostic digital twins for the participants in a clinical trial. And we're also going to evaluate it. And the method by which we evaluate it is important because the predictive performance of the model directly feeds into the next step. We need to now account for that prognostic model while we're estimating the sample size that's required for our prospective study. So the better predictive performance we get, the smaller the prospective study can be. And the worse the predictive performance is, the larger the prospective study needs to be, with the limit as just not doing this at all. So if the model is totally worthless, you end up just running a regular RCT, one-to-one randomized, it's with the number of patients you would use anyway. So that's the worst case scenario. And then the last step happens at the end of the trial, in which you're going to estimate the treatment effect from the study using a generalized linear model to adjust for the participants' predicted control outcomes. Now, in the simplest case of a continuous outcome, which is the case, this specific case that's covered within our current EMA qualification opinion, okay, PROCOVA becomes a special case of another statistical technique called ANCOVA. But with something like a smart covariate, it is not just a simple covariate like age or weight that you would measure when a patient enrolls. Instead, we're going to take all of that information and pass it through this computational model to construct what is essentially the best possible choice of covariate. And we can think about looking at this in the following way. We're going to make a plot, and along the x-axis, we're going to put the predicted outcome from a patient's prognostic digital twin, so the predicted control outcome. And along the y-axis, we're going to plot the observed outcome for the patients who are actually receiving the control. And then we can think about the relationship between the predicted and observed outcomes. If the model were perfect, then everything would just fall along this diagonal line, which would have an intercept of 0 and a slope of 1. But in practice, our model is not going to be perfect. It's a statistical model that is learned from data. So the relationship may be different. We may have an offset alpha that is not equal to 0. We may have a slope beta that is not equal to 1. And we may have some r squared, which is less than 1, maybe a lot less than 1, in fact. And that's describing sort of the strength of this relationship. And one of the important, really the most important properties of PROCOVA is that when we think about the bias in the AI model used to create these prognostic digital twins, that manifests in alpha and beta. And when we use PROCOVA to estimate the treatment effect at the end of the trial, neither alpha nor beta impact the analysis in any way. So any bias that we have from the model does not lead to bias in the resulting study. The only thing that matters, in fact, is the r squared, which is used for estimating the sample size at the beginning of the study. So that's maybe a bit of a technical description, not super technical. If you want that, you can read the scientific papers that have all of the theorems. But a basic analogy, because people often still ask this question. They say, well, you know, I hear in the news all the time about artificial intelligence and machine learning and statistical methods creating biased predictions. And that bias that they have, that can impact people negatively. We hear about this constantly. If we're creating a prognostic model to use for clinical decision support, for example. And that model makes incorrect predictions that could lead to patients getting the wrong treatment. And we don't want those things. So there's this constant tension of how do you create models that minimize that bias and find ways to use them and ask, well, when I'm using it in such a way, am I going to potentially have a negative outcome to people because of the bias? This is a constant thing in the news. And in our case, we can think about this in a very interesting way. So let's imagine this again. So it's kind of maybe a dumb analogy, but we have a Trojan horse. Our AI model, it potentially has learned some bias from this historical data. So we're using this model to create these prognostic digital twins. They could be biased. Okay, and now we have this application where we want to estimate the treatment effect, the average treatment effect from some clinical trial. And the concern is that our Trojan horse is going to somehow bias this application. That's going to bias our outcome. And by using a randomization and PROCOVA, we can essentially create here like a moat that protects our application from any bias in the model. This is a very special thing that we're able to do in randomized controlled trials that you can't do in other situations. We are able to use randomization as a shield to protect the outcome of the trial from bias in these models. And that's a really interesting and important aspect of this application. Okay, now, just because we say bias in the models doesn't matter, doesn't mean that the performance of these models doesn't matter. The predictive performance measured, say, as that correlation absolutely matters. The more and more predictive we can, the better and better we can predict the outcomes of patients on control, the smaller we can make the control group in the prospective trial. So estimating this correlation or discrimination and other types of endpoints ahead of the trial is a critical piece of what we have to do, and we need to do this carefully. So as we get better and better correlations, the size of the control group gets smaller, as you can see on this plot. And we've been working with the EMA to obtain a qualification opinion on PROCOVA, in particular for continuous outcomes. So for the special case that looks like ANCOVA. And we started this process in April of 2021. We submitted an application to the EMA for a novel methodologies qualification opinion. Had a number of different meetings and interactions with the EMA over the past year. And then in March of 2022, they published a draft qualification opinion. In this draft qualification opinion, describes the suitability of PROCOVA for both phase 2 and phase 3 clinical trials with continuous outcomes. And in particular, describes that suitability, not just as supporting information or for secondary outcomes, but actually for the primary analysis. So this is a method which should allow sponsors to run clinical trials with smaller control groups at both phase 2 and phase 3 in a way that the regulators will actually accept as evidence. So we can summarize this pretty quickly on a simple 2x2 matrix. Along the horizontal axis to the left, we have trials that require a larger number of subjects, larger number of participants, and therefore enroll slower. They take longer to reach total complete enrollment. And on the right, we have trials which are smaller. You don't have to enroll as many patients. So therefore, you can reach total enrollment faster. And along the vertical axis, we have things at the top that produce really reliable evidence. And along the bottom, we have trials that produce less reliable evidence due to confounding. Now we have a traditional large RCT, which requires a large number of participants to produce a sufficiently accurate estimate, a sufficiently precise estimate, of the treatment effect. Then we have TwinRCTs that are just as reliable as traditional RCTs. They produce the same exact kind of evidence. You can interpret it in the same way, but they don't require as many participants. So they're faster. And then down here in this bottom right-hand corner, we have this other idea of running trials with a simulated control arm. So this would be a single arm trial. Every patient receives the experimental treatment. So those trials require half as many patients, right? So they're way faster than a traditional RCT, but they also produce less reliable evidence because we can't be sure that there aren't some unknown unknowns that are causing us to come up with the wrong answer. So before moving on to take some questions, I just want to recap the various things I've talked about today. So first, we described a prognostic digital twin of a person as a computer model that predicts the potential health of that individual person in the future under some conditions. And at Unlearn, we create these prognostic digital twins by using deep learning applied to these large compendia of historical patient data collected from previously run trials, observational studies, and some real-world sources. We apply these prognostic digital twins within what we call a TwinRCT, which is a randomized controlled trial that uses digital twins of the study participants to enable designs with smaller control groups that still provide high power. And typically we're talking about somewhere between 20 and 40% reduction in the number of patients who are required for the control arm. That varies depending upon various aspects, and it's one of the first steps in the partnership, and it's really actually step 2 in the PROCOVA procedure, is to estimate exactly how much you can reduce the size of your control group. And just to finish off, to remind everyone of the value proposition that you get from using TwinRCTs. You get to run clinical trials that have smaller control groups. So they're smaller overall, they enroll faster, but I think very importantly, they are more attractive to potential participants because they have a higher chance of receiving the experimental treatment, which is the #1 reason that patients give for wanting to participate in clinical trials in the first place. You get to do that while still maintaining high statistical power while controlling for bias. And in the end, you generate evidence that is suitable for both primary analysis in phase 2 and phase 3 clinical trials. So if you have questions that we don't get to with Arsalan here in the Q&A, feel free to reach out to me at charles@unlearn.ai.
Right. Thank you as always, Charles. Very informative. Love your presentation style. Let's get to the questions over here, and I will do as my best to bucket these up. Let's get to this first one over here from the audience. So I'm going to read the question just as is for the audience. Does the model assume stability of health futures? If yes, how does this align with observed instabilities of cohorts seen even with RCTs using all concurrent controls?
So our models describe the potential future of a patient in a probabilistic way. So when we're trying to make a prediction about the future, we can't do that totally confidently. We can't say this person's going to have a relapse in exactly 102 days. That would kind of be that's impossible today. So we're making these predictions in a way that describes a range of possibilities for the future. So we're talking about uncertain predictions. And that uncertainty can reflect a whole bunch of different factors. It could reflect things that we just didn't measure some of the things at baseline. So we don't know what the patient's age was. So it's harder to make that prediction. And it can also reflect some of these things, which is that because just within the world, there is things that change that leads to some uncertainty in our predictions in the future. The key factor for us is that because of the way we use these predictions within clinical trials, by leveraging PROCOVA, we can ensure that you still get unbiased, valid estimates of treatment effects out at the end of your trial, even if there is some drift like that in the actual patient population.
Okay. Let me ask you a couple of questions because we're talking about the regulatory phase here. So you did mention some interactions with EMA again here. Some questions about, have you engaged with the FDA and are you planning on re-engaging with the EMA? What can you say to that, Charles?
So I'll start with the EMA. The EMA qualification opinion is, we're still discussing this with them because they released a draft qualification opinion, and then there's a comment period. And at the end of the comment period, there's a revision, and that's where we're currently at. So that is still ongoing. We expect that to be a couple of months before it would be finalized, but we don't know for sure, right? So that conversation with the EMA is still ongoing. Will we follow up with additional requests for qualification to EMA? Yes, almost surely. We have a number of, like I said, this is a family of techniques. We have things related to that are not covered by this, related to how do you do things for binary outcomes, yes or no questions? How do you do things for time to event outcomes like survival? There's a whole bunch of range of different variations on that. And yes, we would like to get those types of things qualified as well. As far as the FDA, we are engaged in discussions with FDA. The pathways for qualification and review of methodologies like this that the FDA has are new, and they're not quite as mature as the EMAs, which has been around for years, and a lot of stuff has gone through it. So the EMA process is like they give you a schedule and you meet with them on these months, and then you get to the end. And the FDA's process is just kind of more open-ended. So we're engaged in those discussions, but can't say for sure when they would complete.
Okay. Question from the audience here, I think is a direct one for you, Charles. Can you elaborate how you generate smart covariate from available covariates?
Sure. So it's actually kind of an intuitive idea. You can prove this mathematically, and we have a whole bunch of theorems about it. But any covariate, so we have some covariate that we're going to adjust for. The amount of power that I gain from doing that is related to the correlation between that covariate and the outcome. So you have this then question, it's actually an optimality question. What is the maximally correlated thing with an outcome? And the most correlated thing with that outcome is the outcome itself. So if you could predict that outcome, that should be, if you have a really good predictive model, that is the maximally correlated thing. That's the thing that gives you the most gain in power. So since what a digital twin is, a prognostic digital twin is a prediction for the outcome, that is essentially, that's not just a smart covariate, it's the smartest covariate. It is the thing that gives you the most gain in power.
You emphasize the word large when describing the training data sets. Can this be quantified in some way? It would seem that a reasonable amount of potential prognostic variables would be required along with the clinical outcomes. Is this viable in rare diseases?
We can't necessarily say a priori how big of a data set you would need, because machine learning is a very empirical science. We really just have to test it. For a lot of the areas, so the size of a data set, the way we think about it, the number of samples is going to be the number of participants that you have times the number of follow-up visits. So how often, how many times have you observed data from each one of those participants? And so that number, the number of people times the number of time points, in many of our cases, we're talking about like 100,000 subject visits. And then the number of covariates that you're measuring, the number of features might be 100, order magnitude 100, somewhere like that. So you're talking about like a 100 by 100,000 matrix of information. If we could get them bigger, and over time they will, they're continuing to grow. We're continuing to add to our data assets all the time. So we anticipate those getting larger. Now the question on rare disease is an interesting one. Again, it's an empirical question. We have models that actually perform very well for diseases like ALS, which is a relatively rare indication. Does that mean we could do this for every rare disease? Probably not. There are certainly going to be, any machine learning problem needs data to learn from. If there are no data, you cannot use machine learning.
Well, that actually leads me into another question I have here, which is what disease areas do you plan to expand into? And maybe you could give us what disease areas are you quite confident in?
Sure. We are currently working in neurosciences and immunology. So we have built models for a lot of the major neurodegenerative diseases like Alzheimer's and ALS and Huntington's, and are also working on Parkinson's and MS now. We are starting to expand into immunology, and the intro you mentioned our partnership with Merck KGaA in Darmstadt, Germany. That's around applications of this approach in immunology, and that's another area we're looking to expand a lot in the next year. Over time, outside of that next year, we do plan to expand into other therapeutic areas as well. And we are open to partnerships where we do that now. We have a structure where we can actually do this in a way that we can test this on new disease areas in partnership with a pharmaceutical company. So if that's something that you're interested in, we're happy to discuss what that could look like.
Okay. Well, let's talk about partnerships actually, because you, like you said, you did mention that partnership with Merck KGaA in Germany. Do you work with, are you only working with sponsors, IP holders? Are you working with CROs too? How does that look like if you work with CROs? I mean, do you work with CROs?
Currently, we don't. No, I mean, it's not that we don't. Like, obviously, they are involved in the partnership when we're working with a sponsor, but our contracts are direct with sponsors today.
Got it. Okay. Now, here a question from the audience. How might this technology be used in traditionally large and long duration studies like MACE?
So this technology should enable more efficient clinical trials generally. Again, provided you have data that you can learn from, right? So provided you have data, historical data to learn from, this type of approach can be applied to enable more efficient clinical trials across the board. And that could be, so, you know, even like these crazy large cardiovascular trials that have huge numbers of patients, these approaches can definitely be applied there. We haven't done that today. As I said, we're focused on neurosciences and immunology right now, but applications into across disease areas are totally feasible.
When you're designing a TwinRCT, how do you determine the maximum reduction in sample size?
So the maximum reduction in sample size depends on the predictive performance of the model. And there are a whole bunch of different factors that go into it. For the main source of truth for this, I would recommend we have a handbook that accompanies our EMA qualification opinion, the draft qualification opinion. And that is published on the EMA's website that describes kind of in a lot of detail how to do this. So what I say here is not what's written in that thing, probably because it's coming off the top of my head, so I don't want to mess it up. But the basic answer is that you are going to have this pre-specified model before your trial starts. And we're going to need to estimate the performance of that model within a trial, within a population that looks like the patients who you're going to enroll. And we're going to estimate, like, you know, what's its R squared or something like that for a continuous outcome. And then there's a formula that we have derived that converts that number into a sample size. And now that sort of gives you like the like a minimum sample size that you can use for a trial. There are other considerations that go into sample sizing, from secondary outcomes to safety. So of course, in your discussions internally and with the regulators, you have to go through all of that thinking. But the minimum sample size would be determined by that formula. And again, the best source of information is the handbook that accompanies the draft qualification opinion.
A question from the audience. Going back to the contingency you mentioned earlier, do you have an estimate for what percentage of conditions or sponsors have the necessary historical data, and how limiting is this need, Charles?
So we have our own historical data for a number of diseases already. We have a group of people, a team who is going out and building these data assets through, well, sometimes we just buy them. Sometimes we are collaborating with academic researchers or nonprofits. And sometimes they are through partnerships with actual clinical trial sponsors. So it depends, this question of how much data there is for a particular disease is something that we definitely explore all the time. And again, it's something that we can do in the very early stages of a partnership. So if you're interested, we have people come to us with interest in working in different diseases all the time. The truth is sometimes we tell them no. We do tell people no, that we looked around and it doesn't look like we can find a lot of data right now. So I'm sorry, I don't think we can help you. If we can't help you, we're not going to waste your time. But the very first step, is we can go spend, 2 weeks or so and do, a literature review to find potential sources of data. And if it looks like it's promising, then we can move forward.
I'm going to ask you this question here directly from the audience because there's several, but they're touching on pretty much exactly the same thing. But I'm going to read this question that just came in from the audience and have you respond to it and answer it, Charles. Do you consider traditional RCTs as the gold standard method? Question. If so, have you compared results from TwinRCTs to traditional RCTs for the same or similar trials, and what do you observe?
Yes, we consider large RCTs as the gold standard method. We can mathematically prove that TwinRCTs produce the same statistical properties as those large trials. So for any individual trial, there's variability, and that's certainly true. But on average, they produce exactly the same type 1 and type 2 errors. They have the same statistical properties. And we do this all the time with pharmaceutical companies. So step one of every single partnership that we do is usually reevaluating some previously completed clinical trial before we move forward. And there's a whole bunch of reasons why we do that. One of them being this question about how do you actually measure this R squared when you're going through to do your sample size estimation for a future study. Well, a great way is to look at a previous study and to actually see what it was. That's one of the great ways. So every partnership looks like that. But it's a really interesting case. There's not that many products in the world where you have a mathematical proof of its properties. And that's actually something that we have here. So not only do we have empirical evidence from simulations and from case studies, we actually have real formal theorems that describe its properties.
All right, we're just about our time here. So I just have one final question for you, Charles, here. So we've heard that TwinRCTs can reduce the size of the control arm. But in your opinion, is there a future in which we can run single arm trials and replace the control arm with digital twins?
Is there a future? So everything here, in my opinion, depends on the timescales on which you're talking about. I believe in the future, artificial intelligence will do everything. if you're talking about hundreds of years in the future, that's a difference. That's one thing. Now, if you're talking about 5-10 years in the future, then I think that for most phase 3 clinical trials still need to be randomized trials. I do not think that people should be trying to run single arm trials with any kind of external control for most phase 3 clinical trials today.
That's awesome. Well, that's going to be the last word, Charles. I always enjoy having you in these sessions, and we always learn so much at Endpoints from when you come up here and do this. And that's all the time we have for today. Thank you to everyone in the audience for tuning in. Thank you to Unlearn for sponsoring this excellent panel one more time. And if you'd like to rewatch this webinar or share it with your colleagues, a link will be made available tomorrow. I'm Arslan R for Endpoints News. Thanks again for joining us, and we hope to see you at a future event.